I guess at this point, we'll start with introductions and then can kind of get into the presentation if that sounds all right. Uh, so my name is James Keach. I'm the ornamental extension agent as well as being the coordinator for the Master Gardeners over on the island of Hawaii. I'm joined today by Russell Galante, the ornamental extension agent for both Big Island and Oahu right now. And then Hannah, our ornamental extension agent for Maui and Molokai and Lanai. It's my distinct pleasure today to introduce Dr. Maureen Fitch. Maureen Fitch is really well known across the state for work in agriculture.
See, I'm pretty old.
I was going to say experience, tons and tons of experience.
Yeah, this is old. I've been around a while. It's okay. It's fun. I still like it.
And I was going to say, you were one of the people that I'd heard about even before I moved to Hawaii 15 years ago originally. I'd heard your name in terms of tissue culture. I heard your name in terms of all of your kind of cutting edge work with embryo rescue and anther culture. And for folks who haven't had the pleasure of reading Dr. Fitch's work, she's been instrumental in a lot of agricultural advances within the state in a variety of crops that I kind of papaya, sugarcane, some with anthurium, taro. She was instrumental both in producing the genetically engineered papaya, but then also in developing the new non-genetically engineered papaya ring spot resistant papaya.
That didn't go anywhere.
But it was still, I don't know. Technically, I thought it was an incredible advancement.
You run out of money.
That is the sciences for sure. And yeah, Dr. Fitch has published a ton. She has over 50 articles, over 2,000 citations.
Oh so my, I never counted. I like to publish. I'm waiting for approval on this one manuscript.
Oh, excellent. We'll have to keep an eye out for us. Please let us know.
Actually, you'll see a bunch. In fact, two of the pictures on my first slide are from there. So I hope it gets accepted.
With that, I think this would be a great time then. I'll open it to the other agents. Russell and Hannah, if you have any other announcements about this, but otherwise, Russell, I see you both have been muted.
Yes, thanks for the introductions, James. The housekeeping stuff I will handle. Please stay muted unless Dr. Fitch asks for you to answer questions or ask questions. If you have questions that you want answered, please put them through the chat. That's in the bottom of the screen. There's a little icon that says chat. You can send your questions through the chat. We can open up at the end for conversation. Also, I'm going to send out towards the end a survey. It's going to be a demographic survey. It's a link to the survey itself. Please fill it out. It helps us for reporting. And then at the end, after the talk, we're going to also send out another survey. So just be on the lookout for that. And other than that, I think that's covered.
Anna, did you have?
Yes, thank you for the introduction, James, and housekeeping items, Russell. If you are here for Landscape Industry Certified Technician CEUs, please, you have to fill out the post survey. So please fill that out so we can get you credits. And you can also email me. I will put my email address in the chat just to verify that you're here today. But please make sure to fill out that post-webinar survey so that you can receive your LICT CEUs. Thank you.
Thank you, Russell. Thank you, Hannah. And with that, I think there's nothing else but to let you shine, Maureen. We're looking forward to hearing your presentation.
Okay, let's start. Linda said it's okay. The PowerPoint is okay, so I trust Linda. Russell had asked me several months ago to talk about the science behind tissue culture. And what we really want to do with tissue culture is to grow businesses in Hawaii to make more industries. So this is my first slide. I said earlier that two of the pictures are of papaya with roots, and that was really hard to get, but we did it. And the little guy on top is a bunch of tea leaf. It'll turn into maybe 6 tea leaf plants from which you can make huge leaves and make lau lau. So the science behind tissue culture is also called cloning, micropropagation, and it's just a method that we do in bottles to clone plants or animals or bacteria. And yeah, you can make more viruses by growing your plants, but you can also rid plants of viruses, which is kind of a nice trick. So how do we start? On the right of my slide, I have something I pulled off the internet just to show a typical plant with a shoot apex, which is what we want to get. We can also get those little buds in the axillary buds, but we go for the shoots. And yeah, we want to collect it and put it on some special nutrients so that this little shoot can make many, many shoots. And because it's got sugar, food, chemicals for cell growth and division, and this is we feed it and we give it more than it should have. But the nutrients also feed a lot of other things: viruses, fungi, bacteria, spiders, and you don't really need a jar of spiders. So step one is to get rid of the pests. And we normally do this by rinsing a material in running water, wastewater, soak it in dilute bleach for 10 to 60 minutes. We use heated tools to remove the bleached dead parts from the shoot. And this is all using our tissue culture material. And then we cut off a tiny shoot. If it's virus infected, we get something smaller than the size of a common pinhead. A lot of times that has no virus in it, and that's what we would want. We would try to do that with papaya. Linda's done it with taro, and it worked really well. Anyway, we put this shoot on nutrient plate, like we have on the right, and seal it, grow it, and check it. You have to check for the pests. So all you need to start making more tissue cultures is a few clean shoots. So you can have a lot of things that's contaminated, toss them. All you need is 1 or 2 or 5. You would cut that piece into many pieces, spread them onto many plates or jars or boxes of nutrient media. The thing is to spread it out and make more. We have an example. We had a grant from the city and county of Honolulu to make green tea leaf plants for the lau lau industry. My friend asked me if we could do it. And so we wrote grant proposals and eventually got something. We started with 10 little plants from our friend Mike Meltzer, the virologist at the university, and Hart made 300 of these plants in 6 months, which I guess is fine. We had a distribution day in June, and we gave the farmers the 500 plants that we had promised them at the end of this 6-month period. 300 plants were from tissue culture, 200 were from rooted cuttings, because we had this double-edged sword of how to propagate material for growers. And right now we've got another 700 tea leaf plants, green tea leaf plants for these growers, because actually they put down some money to support us. And so we'll pass these out maybe in 3 or 4 weeks. So anyway, the gist of this is once you have some clean plants, you cut them up and you put them into more media. So you do step 7 over and over and over again. A lot of times you'll have problems with contamination, but if you have enough stuff, you can live with it. So why do tissue culture? First of all, we met somebody who had a very rare, valuable, desirable, variegated aroid, and this person wanted it cloned. And so we said, sure, we'll try. using the techniques that I talked about. In another case, and this is something we're doing right now, the papaya farmers have been planting seeds forever. And in the F1 hybrids that we use, Rainbow and Laie Gold on Oahu, the seeds are made by crossing hermaphrodite onto female. And what you get from this cross is half of the seedlings will be female. And the farmers don't want that. They want the nice light bulb shaped hermaphrodite papaya. So they plant many and then they have to get rid of the wrong sex ones, unfortunately, which are female and they're nice and round and fat like a lot of us are. But we can clone papaya hermaphrodites. And if we plant, give the farmers, sell the farmers these plants, in 2 months, they will bloom. In 8 months, a farmer will have fruit to sell versus 12 or more months, you starting from seedlings. And another reason to do tissue culture is exclusivity. If you have something really rare, kind of like this rare valuable aroid, and you want to be the only person who has it to sell so you can keep the price up, you try tissue culture. We have a new tea. We have anthurium hybrids that are knock your socks off beautiful. And we're trying to clone those. So what do you need? You need your special plant, yours, or a wall flower like I'm showing here. This is from Mr. Stanley Oishi, and we're cloning this for his son, Calvin. You need a laboratory, a room in your house, a back warehouse, where you can collaborate with people like us at Hark. We have a huge laboratory. We're set up for everything, and we'd like to help. You need trained workers, UH students, technicians. Right now, we have a Leeward Community College student from our friend who's a professor there, and he comes as a part-time volunteer, but I am trying to get him hired for part-time at our lab. It would be good to have a business plan, an MBA, or at least knowledge of your customer market. And then you need a marketer. Okay, the equipment you need, this is the pricey part. You need a clean air workspace. They're called laminar flow hoods. They send filtered air toward the worker so that you don't have bugs flowing into your cultures and bacteria and fungi. You need a large pressure cooker and autoclave so that you can make aseptic media so these bugs won't grow up in your food. You need nutrient media recipes. You need scales, balances to weigh your chemicals, or you can buy them pre-made, and that's a lot easier, but it's pricier. You need glass and or plastic containers to put your media into and autoclave it. And then you need tools. You need to decontaminate them. We use either glass bead heaters, or I'm a fire guy, so I use propane burner and tanks. Now I'm old-fashioned, but for me it's fast and it's not too expensive. Finally, it's good to have lighted grow racks for the cultures in your lab. And because I'm messy, I put my cultures into double Ziploc bags so that we don't have all kinds of bugs flying into the containers. So Sandy Bear, our friend on the Big Island, sent me this picture of her beautiful lab. It's really clean, it's really neat. Mine is not this pretty, but she has a successful tissue culture business on the Big Island. And probably a lot of you know her. Probably a lot of you have purchased material from Sandy. So how to start? Read. It's always good to read. Talk to experts, tissue culture people, first of all, UH, PBARC, HARC. Check websites. for local, national, international tissue culture labs that sell services. A lot of time, the international ones are less expensive because their labor costs are lower. So you have to figure that out. You can hire experts to do the job. Sandy Bear, Chitose Takami, both of these ladies have their own tissue culture labs on the Big Island, as does Mr. Lee, Thomas Lee, and Hart and other places. We tend to write a lot of grant proposals. Occasionally we get something funded, write them with private labs, with university, PBARC, and Hart can help you with that. And we're always happy to help growers. The benefits of tissue culture are that the plants are provided with all the nutrients and hormones so they can grow faster. You can propagate faster. They're protected from pests, bad weather, disasters like the volcano. You have exclusivity. You can control your product and your market. Don't make too many, don't make too few. Do what you can. And finally, you have a worldwide marketplace. You don't have shipping fees to national markets. You are required permits for international markets, and those are not expensive. There are nominal shipping bulk charges, but you can check into that. The problems with tissue culture are that the plants are tiny, they take long to grow, but you can make plenty. You have contamination always, and you have to calculate for that and make sure you have enough clean stock material to back up some disaster. The laboratory is expensive and labor cost is expensive in Hawaii especially. You need a shade house or greenhouse, some holding area for your plants. But most people have figured this out. I want to talk about some heart tissue culture projects that are ongoing. We have sugarcane clones that are being made, even though we have no sugar industry in Hawaii. Louisiana has sugar farms, and we have shipped clones to them at some regular rate, and they seem to be happy with Heart's product. We are also studying projects to try and play with sugarcane and take out some genes that may be dead to sugarcane, like maybe the flowering gene. Bananas here are made for Oahu growers, and those came through collaborations with the university, CTAR. And some local people come over and clone stuff for us, but bananas are pretty difficult to work with, and you can't make too many cultures from one corm that they supply you. First of all, we have to check it for bunchy top virus and other bad things. And if it's clean, then we can start. Some banana clones, varieties just don't grow, so we give up on it.
This is speaking now.
Yes.
And this
I'm sorry? Okay, we have Xanthosoma, which is a relative of taro. Linda has handled that. There was a request to clone that, and she's done it for the industry for taro, a kind of taro chip. We clone anthuriums to growers, sell them or give them with grants to growers. I have this crazy blue anthurium gene project, this genetically engineered anthuriums to make them blue or purple. We'll see, it's still ongoing. It's back burner by now, but we have a grad student working on this. And then we clone papayas, tea leaf, anthuriums, sweet potatoes, rare aroids, stevia, kava, and succulents. And we propagate those for sale. Stevia, sugarcane, and papaya are used in CRISPR projects that we want to take certain genes out and make perhaps a better product without the product being GMO. And yeah, GMO has been a problem. Two examples of current tissue culture projects are from 2020, 2021, HARC was awarded tissue culture products to help growers get many plants quickly. Hermaphrodite papayas and green tea leaf plants with reduced virus. Grant funding came from USDA, Hawaii Department of Agriculture, the city and county of Honolulu, private businesses, and the farmers themselves. And thank you, everyone who supports us. We beg for many letters of support from you, and you guys have been great. Thank you. So I'll talk a little bit about the reduced virus green tea leaf plants for Hawaii growers. Mike Meltzer made these. He's a very clever guy, virologist at the university. And with his help, we got a city and county grant. So from January until this past June, we had this timeline to make 500 of Mike Meltzer's very reduced virus green tea plants. We're not calling them virus-free because Mike says there are probably 2 other viruses in there besides the 5 known. And since he doesn't know what they are, we don't have tests for them. And so we have to say it just doesn't have 5 of the 6 or 7. So they needed larger, healthier green tea leaves for lau. Tea is a canoe plant that came with the early Hawaiians in the 700 ADs, but it has 6 or 7 viruses because it's been around a long time, and these plants can collect a lot of these viruses. The leaves become spotted, yellow, and too small. Leaf size was decreasing. Dr. Meltzer used tissue culture and his anti-freeze technique to make reduced virus green tea plants with large, clean leaves. And the farmers heard about these, and they want to supply Keoki's Lau and Ono Ono and other places which make Lau for Hawaii, and they also ship to the mainland markets where Hawaii people live and know about Lau. So, as I said, we did it with city and county funds, and farmers seem to be happy with our work. On the left, you can see infected lau tea leaves, and the leaves are getting smaller and smaller. So you couldn't wrap a lau with that little thing. It has to be 19 inches in length. In the center are some of these clean plants from Mike Meltzer, and on the right, some growers in a clean field in Waimanalo on this island. So we started with material from Mike. We got 10 tissue cultures, little plants in boxes, like you see at the bottom there. And so we took these plant clusters out and we did the chopping and grew them on gel, and we made 300 plants for the growers, which is cool. Mono, our greenhouse fellow, is 6 feet 4 inches tall, and that's him in the greenhouse with some of the cuttings that they were propagating in the greenhouse and in the field. And below is a little tray, a 10 by 20 with 2-inch pots, 50 pots in a tray of the tissue culture material. And in the greenhouse to the left of Mono, you can see the little plants that went from 2-inch pots to 4-inch pots. And farmers seem quite happy with that. And this is us on distribution day at a farm in Kahalu. And there's me with the happy farmers. Myron Murakami, the fellow in the blue shirt in back of me, he's the one who engineered this whole project and made it come to fruition. He got money for us, and he's our good buddy. So this is my gang, the Heart Gang, the 4 field guys, tall one, and the shorter ones to Linda's left, right, your left. And the tissue culture ladies on the right. So that was a happy day. I wanted my whole gang to see what a tea leaf farm looked like, because this is where the products of what they do went. And I feel this is important for our workers to see that someone's actually using their material. The other project I want to discuss is hermaphrodite papayas. The growers prefer hermaphrodites because you can stick more in a box and they are more uniform in size for the growers, and they get rid of all the fat females or the little ones that don't, if they don't get pollinated, they fall off or they just make a small fruit, and that's not practical. So we start rainbow papaya tissue culture with a hermaphrodite, and this photo is supposed to show little hermaphrodite buds in the field. We remove the top of the plant, cut the leaves off, soak in bleach to remove bacteria, fungi, bugs. Then we trim these materials, and we can actually trim the shoot on the top and each of the nodal buds when we put them in tissue culture. Once we get a clean culture, we will grow a little plant with a callus. We learn to chop off that callus and stick it in rooting hormone and stick it in a jar. The roots come out. We stick it in vermiculite medium. And with lots of roots, we plant these things. And it took us a while to figure out how to do all this stuff. But now it's working and we're really pleased. We can make hundreds of plants. Okay. 2 months in the bags. And somehow they're in dryish soil, but there's a lot of soil moisture inside. So all of this comes from the plant. We're not adding any more water. It's fascinating to me. And this is our group. We did both rooted cuttings of rainbow and microprops. And the tissue culture guys are shown behind the plants the day of shipping to our grower on the Big Island. And that was a happy day in August 2020. And so with HDOA, Agri Business Development Corporation grant, we got $7,000 for them to make 700 plus rainbow hermaphrodites to give to one grower who was impacted by the volcano in 2018. And we chose, Sharon Wages and I chose Orlando Manuel because he's our friend. He's a good farmer. So he was selected, and we shipped these papaya plants over mid-August. And this is the plants ready to go into the field by the end of August. After 2 months, the plants had buds in the field at Orlando's in Opihikau. And after 8 months, he had fruit to sell, ripe fruit in 8 months after planting. He said, this is the way to go. And he's very pleased with the product. New heart tissue culture projects include the Tea Leaf 2022 grant for green and ornamental tea leaf. This is for Lala Landscape, Floriculture, and the nursery business. So if the client has a pretty plant, ornamental, like our friend Bob Osgood, we are going to clone this for him starting in January, but we already started. We're practicing on this Kauai Beauty. That is a very pretty ornamental tea leaf. And then his hybrid, the 2018 hybrid that he hasn't named yet, it's got these neat dark purple, red margins. So we'll be trying to propagate that for our friends. So that was the end of my talk, but I want to place this question in front of you. So where do we go next? Russell's my friend, and we're thinking of having virus-free red ginger and other colors available to you growers. Years ago, Linda made virus-free taro very well using tissue cultures. Taro has a Dachine mosaic virus, DMV, and she recovered clean plants after making little pinhead-sized shoot tips. And she grew them in culture, grew them up, kind of like Mike Meltzer did with his tea leaf, tested them for this one virus. They were clean. And so she was able to give plants to researchers, to growers, and Hark sold some. So we can try this with some virus-free red ginger that we already have at Hark, thanks to Hannah and Russell. And we can write a grant proposal and maybe get it funded. So
Yeah,
that's about it.
yeah. My 2 cents on that. I think the red ginger and related ginger and the Alpinia purpurata tissue culture project is probably the most important one in the state right now. I might be a little bit more invested than others, but the fact that we have almost no virus-free gingers in the state for nursery and cut flower is a serious problem. And it's
on this island, it's terrible. The ginger is awful. They're all stunted and terrible looking.
Oahu is ground zero, but we've done testing statewide, and there's virus ginger on every island. And those that think they don't have
My question is, how long can you keep them virus free? Because this is not GMO like a papaya. These will get it again.
Well, yeah, but it's a lot like other plants that have virus issues. It takes them a long time once they're virus to decline. Years. So if you
like
start
bananas, bunchy top.
Yeah. If you start clean, it's better to start clean and try to work on not letting them get infected. But once they do, you have several years, if not more. It's a long-term issue. But virus-free is the way we need to go. So if there's any of those tissue culture labs that would want to start up on the project before we get a grant, we should talk to them, because it's definitely a money maker also, because I think people would buy virus-free.
Andy or Chitose or Mr. Lee, sure.
I need to talk with them.
And I'll add for the taro as well. I think there'd be some great interest with that. I know Susan Miyasaka's getting a project together right now on sweet potato, doing something similar. That taro, there's a huge need for taro propagules in general across the state, and if we can guarantee them virus-free, for one thing, we just have trouble moving things across the state because of taro root aphid restrictions. And kind of like you said at the beginning of this talk, one of the great things about tissue culture is it sidesteps all of these quarantines and other issues. So
national and international markets.
I've brought back blessed orchids from Thailand many times, and it's a lot nicer bringing in those than trying to bring in almost anything else and having to deal with the permits. But yeah, for sure, I think all of the work you've described today, there's huge interest, and we can continue the conversation. I guess at this point, are there questions from other folks? You're welcome to put them into the chat. At this point, I think we could also have people unmute if you prefer to do that. Just kind of, if everyone unmutes at once, it might be a little chaotic. So just be conscientious as
Oh, it should be fun.
And for folks who aren't as familiar with Zoom, I know we went through and muted a lot of folks when they entered. Normally the mute and unmute button is in the lower left, I think, and it's a little microphone icon that has a red slash through it. So if you want to unmute, just click it. Or otherwise, if there's questions that come to mind
Russell,
later, you can let us know.
I'm sorry.
Oh, no, please.
Russell, you can share my email and cell phone number with anybody who asks you. No problem.
Yeah. Well, if you're comfortable, I'll put your email up in the chat right now. How's that sound? Everybody's gonna have it.
A lot of big names in agriculture, in the attendees here, so it'd be great to get some more collaboration going.
So here's Dr. Fitch's email, and above it is the demographic reporting survey. This is something new we have to ask every time we have an event in extension, we need to request that you voluntarily fill out this demographic information. It lets us tell the federal government who we're servicing. So it's pretty helpful if you fill it out. Of course, it's voluntary. It's going to take 1 minute at most. This isn't the survey that's going to ask you questions like, did we do a good job on this webinar? Do you want licked credits? That one's going to come after the webinar. But for now, if you want to fill this out, that would be lovely. Thank you. And Maureen's email is below that in the chat. I had a question. Maureen, I saw in the picture with mono that you had pipe benching. How do those work out? Are they pretty sturdy? You had PVC pipe benching. How do you like those?
The benches are those Benchmaster things.
Huh.
Fine, we love them. Yeah.
How tall were they compared to mono? Just so I know how tall those plants were.
Typical height, what, 30 inches?
Okay.
Yeah, mono's very tall, but we love him.
You were using perspective to trick us into thinking those plants were taller than they actually were.
And please come and visit us at Hearth if you're ever in Kunia from other islands. It's fine. We're happy to host anyone, show you around.
Dr. Fitch, I was wondering, I'd heard of heat and I'd heard of some other chemicals. This is my first time hearing of Mike Meltzer's antifreeze treatment. What are your thoughts on that or on other sort of virus elimination?
It's something that is used for cryopreservation of cells. It essentially sucks all the extraneous liquid out of cells. And so the bigger mature cells have they have vacuoles and a lot of liquid in them. So the antifreeze just sucks all that stuff out. But the meristems, the shoot tips, they're very tiny shoot tips, this pinhead part, it's got really dense packed cells that the antifreeze can't steal all that water. And so then you put this stuff in antifreeze, and then you throw your material into liquid nitrogen, which is pretty darn cold, for 45 minutes, according to Mike. And then you take out this stuff, thaw it, and this is how they store plant material long-term in liquid nitrogen or very cold temperatures, so that you don't have to keep propagating and changing the material. Then when you want a little flask of cells, you just thaw it and something will grow. And this is what Mike did. And then he put these little plants that regenerated green leaves, put them in his greenhouse, and he gave some to some growers after he tested the plants. Did not have 5 of these 7 viruses. And this one gentleman in Waimanalo, he was bold, and so he said, sure, I'll try it. So he's had his material in the field for about 3 years now, and he's got 30-inch tea leaf leaves. All the other growers who are trying to get this material through this grant we had, they have little 19, 20, 16-inch leaves and full of spots and yellowing, and so they want this material. So thank you, Mike, for fooling around with cryopreservation, or it's called cryotherapy. And it can be done. There are easier ways we can do this shoot tip, this tiny shoot tips. And Linda would try that if she had to clean up some red ginger. But fortunately, Hannah found some clean stuff for us, so it'll save some time. And we're growing Hannah's material in our clean room. We only allow clean ginger plants in there. And then we have tissue cultured papaya plants, and they don't have any viruses that can hop onto the red ginger and bikini pink. So it's fine. Was that sufficient? Was it understandable?
I learned a ton. Yeah, thank you. I'd heard of cryotherapies, never heard of antifreeze used for it, but that makes great sense. Thank you so much.
Yeah, people are using this in New Zealand, I think, for cleaning up grapevine.
Oh, okay.
cultivars of grapevine.
All right, I see a question in the chat asking about does Hark sell cloning kits, or do you just provide the cloning service directly?
You know, we're cheap at heart. We want jobs. So some people will charge $3,000 just to get started, but we're nosy and we want to see how good we are. So we're not charging anything. But if we have a product, then I think we have to try for this $10 a plant fee that we're charging for the papayas. And when you really think about it, it's a deal. You don't have to grow seedlings. You don't have to multiple plant and wait until they shade each other out and cut them back. But to convince big growers of that, that's tough. But we're working on it.
I can see some real advantages with that. I see another question in the chat about orchid projects. And specifically, it looks like this person is bringing in orchids in vitro from Thailand. What is the
It's
recommended?
way cheaper.
Yeah, what is the recommended clone quantity per shoot before problems start arising? And I assume they're asking about things like somoclonal variation or things like that.
I believe orchids don't have much of a problem. It's not like the bananas that you can only get 1,000 clones from one corm. And actually here, they're only making 400 from one. And then you have to start with another corm. So yeah, if you go beyond this 1,000 from a corm, you get big ones and short ones and fat ones and skinny ones, and it's a mess.
Yeah, I've heard bits of Taiwan too about the research and what combinations result in kind of the lower somoclonal variation, these mutations popping up in orchids. Some really high quality firms there doing some great work.
Yeah, I think you just have to stay away from making too many and using maybe too high a growth regulator combination. But so far with our papayas, our microprops, no problem. We've had some striping on the fruit, and some of the growers say because it's not green, it's not photosynthesizing enough, so they don't want our striped papayas. So we tell them, okay, we send you these plants, you tell us which ones have stripes, and we'll get rid of that particular clone. And we have. No, we've been lucky with papaya. With taro, we got some stunted ones and weird ones, and I think that's just from making too many.
That squares with what I've heard. Oh, I see a question in the chat now about question about concern with virus on alpaca, which seems to be spreading. Have you seen any tissue culture work done on this, or?
No, not at all, but we'd be curious to find out what the problem is, so please let us know.
Yeah, maybe that would be something. I see they ask Hannah in the comment, maybe a chat with Hannah some more about that specifically. Go ahead.
The cucumber mosaic virus is the virus that's affecting the naupaka. And it does seem to be spreading here in Maui, which is concerning. However, I believe there's a wide host range with that virus. So maybe that could be a potential project in the future, is looking at tissue culture, ways to control the virus. Any thoughts?
Sounds good. We're happy to try anything. We start with the simpler shoot tip culture, and if we can clean it up, we can try to, but to do this sort of thing, we do need a grant proposal because we have to pay Linda and me and our technicians. We can do the isolation, but we really need our technicians to take care of the plants in vitro. And we've got 3 ladies, they're very good. And we need to keep them employed. So we're always looking for funding. Thank you. So write to us.
Let me answer, or let me get to some of these questions. I want a chance to talk. My time in the sun hasn't been enough for this webinar. Thanks, James. Okay, Sean asks, I'd love to sell some blue anthuriums. Any idea when those might be available?
Oh, the wall anthurium or the blue anthurium?
Blue anthurium project.
Well, the kid's got to graduate, so I'm nagging him, and I told him, you don't have to bloom it. Just show that the genes are there, and then we will try and grow them up and bloom them. But then we have to go through GMO deregulation. But because you don't eat this stuff, that's a plus.
Yeah.
But
it's a timeline of years. Yeah, correct.
10 years, easy, 10 years.
10 years, okay.
Yeah, but let me tell you this other thing. There's something going on in USDA now. There's a purple tomato, and the genes that are in this purple tomato make a lot of antioxidants in the tomato. So this is a really healthy tomato. If you eat this, you're getting more antioxidants, more anthocyanins than in a blueberry. So this researcher, her name is Kathy Martin, and there was an article in the New York Times about 3 weeks ago now about Kathy's work, and she's a very well-renowned scientist. Linda and I happen to know this lady and some of the anthurium people, because when she published her paper in 2008, I read it and I thought, oh, cool. I'm not looking for healthy papayas. They're really healthy already. But a blue or a purple anthurium, this will be cool. This will help the anthurium growers. So I wrote to Kathy, and she's so nice. She wrote back, and I had asked her if she can share her blue genes with us. So she did, and we have them. And we made some anthurium plants. Some of the material came out positive, but we can't work on this stuff because we're not paid to make blue anthuriums. So it's kind of on the back burner, sitting around. But in the meantime, Kathy's material is going through deregulation at USDA, and she wrote to me in her email that maybe by the end of the year, the purple tomatoes will be available. And she just wants to give them out to growers, to homeowners. Grow it in your backyard, eat the healthy tomatoes, and be happy. She's British. She's from England, but so they can't do anything like this in UK or in the EU, but we can do it here in the US. So it's a hopeful sign for GMOs, that maybe GMOs are not quite dead yet when it comes to human health. And that's a real plus. And I can send anybody this article. I've got a PDF of it. It's kind of long, but the reason I heard about it is because my auntie reads the New York Times every morning online. And on the front page, there was a picture of Kathy's purple tomato and rainbow papaya. And the rainbow papaya came from Michael Cohn up the street from us, who's our good friend. He loves us. And we keep his business going with the Cobalt 60 irradiator. He works with Lando, our farmer friend, Orlando Manuel. And the papaya on the front page of the New York Times is Lando's papaya. So my husband said, well, maybe it's the papaya from the plants you guys sent in August, because he's already selling that stuff. And Michael said that the New York Times called him early July, and they said, we're going to publish this article, and we need to take a picture of rainbow. Can you send us a couple of cases of rainbow papaya? So he did. Isn't that too cool?
Yeah.
And I only heard about it because I don't read the New York Times, right? But my Auntie Jean read about it. And so she called me. She said, Maureen, did you know? I said, no. But then she mentioned the purple tomato, which didn't mean anything to her. So she sent me a copy of the article. And oh my God, how exciting.
Yeah. Yeah, it's great to get in a major media
Yeah,
magazine.
yeah.
We've got a few more questions here. And
Sure.
James, you might need to jump in too, because this is some orchid stuff. So can you keep recloning orchids using tissue culture without any repercussions to, I'm assuming, drifting and
I don't know.
they're asking?
I assume you can, but I won't stick my neck out and say yes.
Yeah.
Reclone our papayas all the time, no problem.
I know it varies a little bit orchid to orchid. We do have a few that if you kind of like Dr. Fitch was saying, if you bump the hormone levels up a little high, you can get some really funky stuff coming out. Some orchids seem to be more
With a lot of
susceptible than
plants you
others.
can. Like the
Yeah.
ornamental tea leaf people, they want us to pump them because they want weird stuff coming out of tissue culture. So
Well,
whatever you want.
and I was going to say, you know, the chocolate orchid that a lot of us enjoy and have in our yards, it all started with, my understanding is one clone, that's, what is it? Sweet cherry, or yeah, sweet fragrance, I think was the long one or something like that. And then there was short cherry that was a somoclonal mutation of that. And then there's all these color variations, and they are all mutations that have come from tissue culture of this one cultivar. But in general, you know, different folks swear different things. I see pretty good results most of the time with orchid cloning.
With consistency.
With consistency and with scrupulous lab use. Like Dr. Fitch was saying, you can certainly get some really funky stuff if you want it by pushing things, but you end up with a lot of orchids that you might have to put out of their misery as well.
Yeah. And we've got one more question about orchids. This is closer to breeding. You know, this individual is asking, if you've got a virused orchid, can you breed it with the same variety and get the same plant, but have it virus free from seeds? Is what I'm assuming the question.
Depending on the virus, you just want to make a hybrid. Some viruses are not seed transmitted. So yeah, you can try. Others are.
Yeah. And I'll say for this too, there's a few factors, like Dr. Fitch was saying, with the type of virus matters. I know for the two big viruses in orchids, Cymbidium and Odontoglossum ring spot, that both of them are not seed transmitted, but it depends on what stage the pod is that when you harvest it and send it in. If you do it as a dry pod, generally you won't have issues with the virus getting transmitted. If you do it as a green pod, depending on how clean they are with cutting the pod, you can sometimes get virus transmission. You will, however, get something that looks different than the original plant. Orchids have a lot of kind of inherent genetic variation, except, you know, kind of harkening back to our local researchers, a lot of the university's orchids were developed so that you could self-pollinate them and end up with something that was virus-free this way. And so if you look at Unui Mist and some of these other orchids that have come out of the university's program, Tessie, Teresita Amore's program, and the researchers before, a lot of those were bred specifically so that you could eliminate the virus while still getting something predictable.
So if somebody's interested in that, they need to look at a little bit of the breeding history of that orchid to see if it is something that possibly is going to have some fidelity in its genetics when feeding it back.
And I'll put my email here if someone wants to reach out about that. It does require knowing a little bit of the background of the orchid and some other things, but short answer, yes, seeds are generally different from the parent, but virus-free. But they do require the same sort of lab setups that Dr. Fitch has described here. You can't generally so orchid seed.
And I had mentioned these three researchers, tissue culture businesses on the Big Island, Sandy Bear and Chitose Takami and Mr. Lee have worked with orchids as well as anthuriums. So they would have the expertise in cloning the different kinds of orchids.
Yeah, I know we worked with Chitose before on sweet potato and on taro, for that matter, when I was in Hilo. Not, I should say, you know, as a CTAR employee, not giving special preference to any one supplier, but we have a lot of great talent in this state when it comes to tissue culture.
We are a nonprofit at heart, so we can be as proprietary as we like. We can sell materials. So,
well, along those lines, we just had a question pop up in the chat about contact info for Big Island tissue culture labs. I know you had a slide earlier with some of the names. I can't remember if there was contact info, or do you want to just name them?
no, but I do have email addresses for both Chitose and Sandy. And Mr. Lee is a friend of Linda, who works with me. And he actually, Linda said, would like to sell his business. But in the meantime, he's still cloning his anthuriums and orchids. So I will check with the two ladies and see if it's okay to share their contact info. And then I'll get back to Russell about that.
Yeah, send it to me, and whoever says yes to giving their contact info, I will send it out in an email.
Yeah, I never give out info like that until I check with.
That's fine. That's fine.
Good
No problem.
practice. Yeah. Other questions for Dr. Fitch while we're lucky enough to have her on here?
I'm done. I'm free. Yay.
If not, yeah.
I can go back to work. Yay. Thank you.
So
Thank
much,
you
Maureen.
for still hanging around. 36 of you. Wow. Bless your hearts. And I'd
You'll
like
have
to
to-
meet all of you sometime. Yeah, with COVID, you know, we can talk on the phone or email. It's fine.
Great.
Please feel free to reach out to any of us. And thank you all for joining us today.
Thank you.
Thank you. Thank you so much.
Thank you. Thanks, Maureen.
Thank
Right,
you.
everybody. So this is our last Floriculture Nursery webinar for the rest of the fall, and we will have the new schedule for late winter, early spring. The next three webinar series webinars will be out. We'll send those out through all of our contacts. So be looking out for those. They're going to be interesting. Aloha.
Thank you. Thanks,
Thank
Maureen.
you, Russell. Thank you, Russell, for this opportunity. It was fun. I enjoyed doing it.
Thanks, Dr. Fitch.
Thank you.
Thank
all.
you. Ahui.
All righty.